Hello, everybody. Welcome to another edition of Biology in the Basement with your host, Mr. Andraco, where we are reviewing all the concepts that we learned this year as if we were preparing for Regents that luckily you guys don't have to take this year. So let's all be happy about that and just enjoy what we've learned here. So let's have a fact of the day so that we can get started. Now, I know you're going to find this joke a little bit cheesy. But it turns out that in 2016, 20,000 pounds of cheddar was stolen from a Wisconsin trailer. And there's actually a suspected ring of cheese thieves. And as of now, they have yet to be caught. Crazy, I know. I hope you caught my cheesy pun there. Anyways, genetics. What is it? Genetics studies genes as they travel through the generations, right? We've tried to do a little bit of wordplay, right? Heredity and inheritance, where we talked about getting an inheritance as what you get from your, family members after they die, when actually we were talking about inheritance as the traits that you get from your family members before you were born, right? Not to sound so cheesy and hippy dippy, but that's what we were looking at, right? Is how we got traits in the form of genes from our parents, right? We are the offspring of our parents that we have DNA directly from, but only half of them, right? That's why we resemble a little bit of both of our parents, but not just one. Some scientists say that up to 60% of offspring, though, do show paternal DNA, which is weird. So it's not exactly 50-50 every time. The first part of genetics talked about Mendelian genetics. Mendelian genetics was the basics. Gregor Mendel was fortunate in finding those pea plants that had a lot of single gene traits, so he was able to figure out things like dominance and segregation and independent assortment in how these alleles, right, can separate and rejoin in fertilization, right? It explains things like why is it that your brother and sister don't look exactly alike? Because the genes independently assort, right? Not every sperm is the same, not every egg is the same. They have gone one or the other of your two alleles, right? Because you're made up of half of your parents' DNA all mixed up, and they are made up of, you are made up, your parents have their DNA all mixed up, right? So it's a good thing as we talk about in evolution when it comes to variation. And so luckily the pea plant helped to show that, because humans and many other species have all these exceptions that we also talked about. He also gave us some great terms that we needed to use, like phenotype and genotype, the physical appearance or the genetic makeup, as genotype is called, right? Your genotype determines your phenotype. Your genotype can be either homozygous or heterozygous, right? Homozygous means you have the same alleles for a trait. Heterozygous means you have different alleles for that trait. But just remember, your DNA determines what your phenotype will be. Of course, as we learned, the environment can affect gene expression. But we'll get to that in a minute. Anyways, Mendel gave us all of this, but he knew that there were exceptions to his rules and questions and things that would help to explain why do we see a red flower and a white flower crossing and we get a pink flower. Well, that's because these aren't completely dominant, right? Some can be co-dominant. Some can be involving multiple alleles or involving multiple genes. And we talked about all of those. And so that kind of helped to explain why there's such a gray area in, two, short people have a tall kid. How does that happen? Well, we learned about that um, because it's just a mixing. Both of their parents had an allele for tallness that ended up in the sperm, and it joined, and that's why their kid is way taller and doesn't seem like they're part of the family. It does happen. The second section of our unit on genetics covered just the structure of DNA. Right? DNA is one of our biological polymers, nucleic acid, along with RNA, made up of nucleotides. We talked about the parts of a nucleotide: 5-carbon sugar, phosphate group, nitrogenous base. They follow the law of base pairing, A with T, C with G. That was always a big Regents question. There are different forms of DNA, right? We just mentioned in the last one, before DNA divides itself, it is in chromatin. Chromatin is when it's in its ribbon-like form. It's about 6 feet long in our cells. And it's like that so it can open up and give directions. But when it needs to move on its journey of cell division, it winds itself up into a chromosome, which is made up of 2 sister chromatids. Then we learned about DNA replication, how it would be very difficult to just build a DNA molecule. So luckily, DNA just opens itself up and uses each open half as a template to assemble parts from itself, so that each strand is 1/2 new and 1/2 the original strand. Couldn't talk about DNA without talking about RNA, because RNA really helps to get things done around the cell, specifically, as we'll talk about in a second here, protein synthesis, another important cell process. But it is a little bit different, and it is only 1/2, and it contains uracil, and its 5-carbon sugar is called ribose. But there are many forms. DNA is just DNA based on the species. But RNA comes in different forms, whether it's a messenger, it's a translator or transfer, or whether it's part of the ribosome as ribosomal RNA. Here are your differences, as I just mentioned. Of course, they do have similarities, right? They're both nucleic acids made up of nucleotides involved in storing and transmitting genetic information. One of the ways they work together is to make proteins, right? Proteins are made with the information from the DNA, head out to the cytoplasm. With the help of the ribosome, we create proteins. It is a two-step process. First, we transcribe in the nucleus, and then we take that message and we translate it out in the cytoplasm with the help of the ribosome. So here is the ribosome translating that message, right, from our messenger RNA into a long chain of amino acids, which we call proteins. The last section of our unit on genetics finished with modern genetics, the things that we learn about in genetics that have helped us through the years, like mutations. We discovered that mutations are changes in DNA, and we can use them for our benefit. Even though most mutations are bad, most lead to genetic defects, disorders, death, but some mutations throughout life have been favorable. And so sometimes we try to induce mutations. We'll talk about that, but most of the time, not good. Anyways, the first thing that we did ever since we've been considered humans is something called selective breeding, where we said, hey, I like this plant, and this plant too here is of the same species. We should breed them together since they're both really good, and maybe we'll get an even better plant. Or this is my best horse, and Joe down the road has a really good mare. We should breed them together, and we'll probably get some good quality horses for our, for our village. That's what we should be doing, and they've always done that. They've taken wild, Mustangs and horses that were nasty creatures and tamed them. Same thing. If you saw a bull 1,000 years ago, they were not kind. Um, the the apple was about this big 5,000 years ago. So through selective breeding, we have accomplished quite a bit, right? We turned a wolf into a pug in 10,000 years. Okay, so think about that. Selective breeding has given us a lot. Um, Now, we've done that through two different methods, whether we hybridize, where we try to take different individuals with differing traits with the purpose of creating what's called hybrid vigor, where if we have two individuals that are different and they cross, hopefully a lot of their good traits will all mix. When we inbreed, we're looking for something specific. This is a really good dog with a nose, and this is a really good dog with a nose. We breed them together, and we're going to have the best sniffing bloodhound that you've ever seen, right? That's why we inbreed it, so that we can maybe multiply a specific trait. Of course, when we try to multiply a good trait, we also multiply bad traits, and so sometimes, as we call selective breeding, sometimes they call it purebred. It can also mean inbred. Um, but if done correctly, pure breeding of dogs and other things can be done correctly to prevent certain genetic disorders. So sometimes we do induce mutations so we can get different combinations of genes that may help us. How do we do that? Radiation and chemicals. Radiation, even simply from the sun, can cause mutation, right? Your skin is a dead layer, because if the internal cells of your body were exposed to UV rays for long periods of time, they would mutate. Same thing with chemicals. If you smoke, if you drink alcohol, if you live in a polluted environment your entire life, okay, these places, these hot spots down in Alabama and Mississippi where there are just no laws about dumping chemicals, and the high rates of cancer that are existing in these communities down there, those chemicals in the ground and in the air, they're causing cancer at higher rates than other places. And so these are the things that will induce mutations. Usually they're cancer. Every once in a while, they're good. Hopefully, maybe Superman will be born from one of those mutants in Mississippi, and they will avenge the companies that pollute. That sounds like a comic book, actually. Anyways, here's my favorite thing to talk about, right? The environment can affect gene expression, right? Where you are going to be a product of your genes, but the environment can change how you turn out, right? Nature versus nurture, as they sometimes say. The classic example we used was the Himalayan rabbit, where they shaved the back of the rabbit, they put the ice pack on, and the fur grew back a different color, right? Just because it was exposed to cold, just like their other extremities. Another example, I think, is geraniums, right? At different pHs of the soil, their flowers will be different colors. So things respond different ways. An example in humans is PTSD, right? If we have gone through an experience where our nervous system has been stimulated to a point where it cannot stand, usually that damages the nervous system and gives it some form of PTSD, right? People who have been to war or have been abused or have just been pushed through limits that they just could not handle. That is another example of the environment changing your gene expression, because it could trigger things in you that may cause you to act out. So the last thing, of course, is second to last thing is, we got to talk about cloning too, is genetic engineering, right? Genetic engineering is where we take the DNA from one organism, we insert it into another to create a form of DNA we call recombinant DNA. And that recombinant DNA usually grows into an organism which we call a transgenic organism, right? Trans, in this case, meaning it has DNA from one source and another transplanted into it. We use genetic engineering to help our crops, right, grow larger yields, be insect and other pest resistant. We use it so that we can have larger fruit that lasts longer on the shelves, more nutrients like golden rice, things like that. Another example that we see is how we have genetically engineered bacteria to produce human insulin. My brother-in-law lives across the street, is a type 1 diabetic, and when he was a young kid, he had to use pig insulin. And pig insulin is not as good as human insulin. Thank goodness for genetic engineering to create this human insulin that works much better and helps him to regulate his blood sugar. So genetic engineering has been helping us. Some other genetic procedures that we talked about and even debated, right, which was a fun time, was cloning, genetic screening, stem cell research, how our knowledge of the cells and what they can and can't do at certain times and certain stages will most likely be carried into the future. But of course, we debated these things because some of you feel strongly for and strongly against them, because you may think that morally or ethically, just or inside, I don't know, that's just not right, right? And so my job is to not just educate you in science, but make you an educated voter. And so we looked into all of this stuff so that you could make your own decision, right? I tried to keep my hands off of, pushing one or the other of these things. I gave you the pros and the cons of both sides, and hopefully you'll do some further research, and when it comes time to vote on stem cells or genetic screening or cloning of humans someday, you will be well educated on it and know what you are voting for, because you're going to get out there and you're going to vote and you're going to be an educated voter, right? Anyways, gel electrophoresis, right? This is how we compare DNA, and we use it for all kinds of things like who's the daddy, who's the suspect, and all of that stuff, where we focus on the fragments and we study the banding pattern. And that was how we ended up genetics. So I will leave you at that, and then the next time we are on to evolution.